I'm Joseph Puglisi, and I'm a professor in the Department of Structural Biology. And I worked on RNA chemistry. And then what I learned is how to apply physical methods to understand structure of RNA. And that's kind of what I've done ever since. There's a class of drugs that are called aminoglycosides, and the one that you all know is, what do you put on your cut when you cut yourself? What drug do you put on?
Neosporin.
You get the answer. Neosporin. Neosporin is neomycin. Okay, it's aminoglycoside. Gentamicin is an example of this. Tobramycin. Right. So these are really important antibiotics, and no one understood how they worked. Okay, we knew that they targeted the ribosome, and the idea, this goes back to the original reason why I work on ribosomes, is that these were drugs that didn't bind to proteins, they bound to the RNA part of the ribosome. Now that gets even more interesting. For about 20-25 years, there was very little activity in the development of antibiotics. In those 25 years, bacteria started evolving. And now a lot of strains of bacteria are resistant to the current antibiotics we have. We have a crisis. As a drug bind to an RNA, at that time we had hundreds of pictures of how drugs bound to proteins. They work in the way I told you. You have an active site where the chemistry of an enzyme occurs, and then you get some molecule that kind of comes in and just sticks in that active site. And like putting gum in a lock, you can't get the key in, the natural substrate in, you get inhibition of the enzyme. But this RNA thing was interesting, and so we worked on that. And what we figured out was how the drug bound to this piece of RNA that we knew was the active site for the drug to bind, and it fit into a little pocket that was made by the RNA. And then what we figured out is, well, in that pocket, the way that the drug touched the RNA was such that if you changed the drug or changed the RNA in that pocket, you would get resistance to the drug. So avoiding resistance is a good thing in drug development. And so how could I practically capitalize on this discovery of the shape of an antibiotic binding to its target? Well, if I was a good chemist, which I've already told you I'm not, okay, what I would do is then go in and try to improve my compound some way, do synthesis of some form or another. That turns out to be very challenging. A lot of these biological molecules that are made by bacteria are very sophisticated molecules. They're very complicated architecture. It takes a really good chemist to make them. So that's not my job anymore. Okay, I've made a discovery. I put it out into the public domain, but other people pick it up. And it turns out there's a company, it's called Achaogen, it's up the street, right? Nothing to do with them, right? But what they did is they modified aminoglycosides to eliminate some pieces of the drug that we thought were not really important for the drug to bind to its site, but were presenting chemical functionalities that could be modified to screw up the drug. So they changed the drug and improved it. And now that's in clinical trials. And we use this to study shapes of biological molecules and interactions of biological molecules. Nuclear magnetic resonance spectrometer, and the large metal cylinder is a magnet. It's a superconducting magnet. It's a giant coil of wire. And what the can is that the wire needs to be cooled by liquid helium so that it becomes superconducting. This is a DNA sequencing instrument. It shines light onto a chip where we can immobilize ribosomes and watch the ribosomes perform their tasks directly.